schedule allows me, when the schedule, and I have it. Today, just lab 1. But what I recommend you do, I have a 3-hole punch in my office, and let me know, come by my office and use it. What I recommend is you get a binder, and just every week, punch holes in it, keep it, and just bring your binder with you. And every week you're going to add on to it. I bought little fun tab dividers that go 1 through 12, so separate tabs. However you keep organized best. What I don't recommend is putting them in a folder, because then what happens is they end up everywhere. So I have a sneaking suspicion that I'm going to So what I usually prefer is us to meet for lecture first. That way I can go over all this this semester. So you see me for lab first. It's okay. We make it work. Okay. So the first technique we're going to learn today is something called aseptic transfer. You can find that on page 29 of your lab manual. The lab manual is not in order, that's why we have the page numbers. And sometimes I skip around for things in lost order. Oh, sure. Okay. So, aseptic transfer. Aseptic means without contamination. And this is something that allows us to keep our experimental cultures what we refer to as pure. This tube says Bacillus subtilis or B subtilis. I know that the microbes that are growing in here are just B subtilis. And I'm going to be very careful not to introduce outside microbes that are not B subtilis into that tube. That way I know when I run my experiments, I'm getting the results for the proper organism. So asepsis is a foundational principle in microbiology. It's also a foundational principle in medicine. It's why you wash your hands before you see a patient. Surgeons wear masks to keep contaminants out of patients' wounds during surgery. All that. In fact, in the history of microbiology this week, you might read about someone who is considered the father of asepsis. And you may have used a product this morning that is named after him, particularly if you brushed your teeth. Something you might do after you brush your teeth. Does anyone know what it is?
Colgate?
No. I hear it. What is it? Listerine.
Oh.
Yeah, Lister. Joseph Lister was the guy who publicized the fact that, hey, we should wash our hands and our surgical supplies before we do surgery. That would really help things out a lot. So they named Listerine after him, which is an antiseptic that people use in their mouths to reduce the number of bacteria. So asepsis just means without contamination. That's just a fun fact you can impress your friends with, depending on how easily impressed they are. So aseptic transfer means without contamination. Contamination can come from multiple places. And this isn't in your lab manual, but I like to let you know where contamination comes from, because if you know where something comes from, you can stop it from happening. So, the first source of contamination by microbes we don't want in those tubes. Any guesses where you might pick up contamination from? The air, yeah, that's one, the air. There's lots of microbes floating around in the air. Fungal spores, bacteria, viruses. Don't freak out, most of them are harmless. The air, our bench tops, all the inanimate objects, doorknobs, all that stuff. Those are considered environmental sources of contamination. So anything from a surface in the environment. So when I show you aseptic transfer, we're going to do things to minimize the amount of environmental surfaces that our experiment comes in contact with. The second, anyone can guess what the second form of contamination is?
From our own bodies.
Yeah. Again, don't freak out. We are covered in microbes. If you're ever feeling lonely, don't worry. You're never alone on a Saturday night because you have millions of microbes in your body that would be so sad without you. We are covered in microbes. We are covered in every surface of our body, pretty much, with bacteria, viruses, fungi that all help keep us alive. We could not live without the bacteria in our guts. You know, the E. coli, Escherichia coli, which we're going to be working with today, a strain of that lives in our intestines, makes vitamin K for us, which helps clot our blood. So we wouldn't be able to live without them. They help digest our food. They take up space so more harmful bacteria can't invade us. Our microbiome is very important to us. Unfortunately, we don't want it in our experiments. So that is considered analyst contamination. And that's contamination that comes from your body. So as you do this aseptic transfer, we are going to not speak because you might, spit a little when you talk. Everyone does. It's okay. You might breathe or cough into your tube. Microbes from your skin might get into the tube. We don't want that to happen. So we take measures as we work to prevent that from happening. The third type of contamination is a little trickier. This has to go with working carefully and in an organized fashion. So this is sample-to-sample contamination. Sometimes you'll refer to this as sample switching. So sometimes when we're in a rush and we're doing things at rapid pace and we have more than one bacteria that we need to inoculate into more than one tube, we might mix them up. And then the results we get for one organism might not be the right ones. So when we work, we're very careful to do one thing at a time. It's not a race. There's no award for the person who gets done first, in lab anyway. Because they bring prizes to lecturers, first team done, that sort of thing. Bless you. So we're very careful to not switch samples and mix them up by working slowly and carefully and with our lab manual as a reference. Bless you again.
Bacteria.
So sample-to-sample contamination is something we can also avoid by properly labeling our tubes. And every tube and petri dish you use in this lab is going to be labeled by you. So that's what your Sharpies are for that you got from the front of your rows and wipe down. Remember, use the lab Sharpies, pens, and pencils to clean your rows. Those are the most common objects that are contaminated. We don't want to bring that back. So, you'll label your tube with your initials. So we know it's yours. We're going to label it with the bacterial and you can abbreviate them in certain ways. So, Serratia marcescens, you can abbreviate to S. marcescens. E. coli is really its full name is Escherichia coli. Staphylococcus aureus, you can say S. aureus. Bacillus, bacillus. You shorten the genus name, you leave the species name alone. I do not recommend using initials like SM, BS, SA, because you might be working with other species this semester that have the same initials, and then it becomes really confusing. If your initials on everything, you need the date, you know how to look. Sometimes you'll also write the name of the experiment. Or the type of media. Which brings me name is going to be one of these when I tell you what experiment you're doing. Oh, so if I'm writing on a tube, tubes aren't that big. So instead of writing out Escherichia coli, I might write E. coli. And you'll see in the week 1 reading, there's a very specific name that we abbreviate microbial names. The genus name, which is this first name here, gets abbreviated to just the first initial, so E, and then the species name stays the same. So Escherichia coli becomes E. coli. Staphylococcus aureus becomes S. aureus, and so on. Yes?
Couldn't multiple genus have the same first letter?
Yes. So that's where knowing what the species is part of the genus comes in. Yeah.
So the species will be unique.
Yes,
Okay.
the species is unique. So there's Enterobacter aerogenes. That would also be E. aerogenes, but you're just supposed to know it's Enterobacter and not Escherichia. Yeah. I didn't say it was a perfect system, but it's the system we have. So what we have here, bacteria and microbes are this is primarily a bacteriology lab, although throughout the semester we will be working with fungi, protozoans. The only class of microbes we don't work with in this lab are viruses because that would require tissue culture and cells for the viruses to grow in. So we don't do any viral experiments. For now, today we're just working with bacteria, so I'll use bacteria and microbe interchangeably. So bacteria are kind of like pets in this lab. We have tubes with them. We have to feed them to help them grow. We have to keep them clean. We don't take them for walks, but they can do some neat tricks you'll see later in the semester. So when bacteria are growing in this tube, they're growing in a substance we call media. So this is clean media. It's sterile. You can see through it. This is media that bacteria has been growing in for a while. See the difference in that bacterial growth is turbid or cloudy. That's how we know that there's bacterial growth in it. Whereas the sterile broth is completely clear. Now this bacteria, this is some Serratia marcescens. So no growth of bacteria. Bacteria, it's cloudy. This Serratia marcescens has been growing in this tube for quite some time. And, when you metabolize, when you grow, you start to run out of room, you start to run out of food, and the waste that you pick up and make as a consequence of metabolism start to accumulate. And no one does well when their room is messy or they're living in their own garden. So, our goal in this aseptic transfer today is to take this bacteria. You're either going to do Serratia marcescens or Escherichia coli, and you're going to feed the bacteria by moving it to fresh medium, aseptically, without contaminating it from anywhere else. Now, throughout the semester, you'll hear me refer to row numbers. Your row number is going horizontally across. So your row 1 is closest to me, row 2, row 3, row 4. I'll often assign microbes by what row you're in. So today, if you're in, the other thing you'll learn about me is I lose the chalk all the time. There we go. So if you see me like spinning up here, I'm looking for the chalk. If you see it, just let me know. So if you're in row 1 or 4, you're going to do an aseptic transfer of Serratia marcescens. If you're in rows 2 and 3, you're going to do an aseptic transfer of E. coli. Now, you're going to do this transfer into 2 different types of media. The first one that I'll show you is liquid broth. This media contains, it's basically like really salty soup, but instead of being made, there are some media that are made of beef and chicken. This media is made of basically soy digest. So it has proteins, it has carbohydrates, it has some salt, it has some nutrients. It's food for the bacteria. It's in liquid form. We also have solid media. This is an agar slant. It contains all the stuff that this has, except it also contains agar, which is a solidifying agent. And we inoculate these 2 different ways that I'm going to show you. So I'm going to demo this. I am going to use Serratia marcescens. And I'm going to demo this now so you can see how to do it. And the way all the procedures in this lab work is we do like the medical school thing. So if you're not familiar with that, it's see one, do one, teach one. We're not going to make you teach one. So you see me do it, then you go do it on your own. And you're ready. If at any point doing this you have any questions, you just call me over and I help you. So the technique I'm going to show you right now is also on or around page 29. There's a picture of it. We're going to do this procedure to minimize the chances of environmental analyst and sample-to-sample contamination. So if you can't see, you are welcome to come up and around. Just don't block anyone in front of you. Your goggles at this point in the lab must be on. Yeah, look at that. Goggles. There you go. Okay. If you don't like the chemistry style splash goggles, like they fog up, you are okay in this lab wearing what I have safety glasses with side protection. That is also acceptable if you find those uncomfortable. Those are, of course, the best protection, the full shield goggles, but these are acceptable as well. So first thing I'm going to do is I'm going to label my tubes. My name is Grace Louise Axler DePert, so my initials are GLAD. The date, 3/6. I always forget the date. The microbe name, this is Serratia marcescens, so I'm going to write S-M-A-R-C, and that's enough for me. It's hard to write on these. There's a little white label on here. Don't write on that. It'll become permanent and we reuse these tubes. Write on the tube. Don't write on the lid. Always hold a test tube by the glass part, not the lid, because if this lid isn't on correctly and you grab it by the top, it's going to fall. You're going to have broken glass and a spill. We don't want that. So did I write everything? I'm also going to write here NB for nutrient. Oh, sorry, that's agar. NA for nutrient agar, just so I know what it is. Because we're going to be setting up a lot of tubes in this lab. Today, that's only 2. But as we go on, you're not going to remember what's in what tube as things go on, so you want to stay organized. So for the nutrient problem, I'm going to do the same thing. The initials. The date is March 6th, 25. The species is S. marcescens. S. mark I'm going to write. And this is nutrient raw. So my tubes are labeled. When it comes time to set up an experiment, this rack will hold the species you need. So some of you will need Serratia marcescens. I'm going to put them here. Some of you will need a liquid drop of E. coli, which is what I'm going to put here. Notice there are only 4 of each, and there are more than 4 of you that need it. So what you're going to do is set yours up, pass it on to the next person that needs it. Okay? We have to share around the lab. So I'm going to start the inoculation. Again, if you need to see, you're welcome to come up. I have my loop. The first thing you're always going to do is hold your loop in your dominant hand. You want as much control as possible. You can come up closer that way. Very awkward at first, but this is the correct way to do it to minimize the chances of environmental contamination. We don't put anything on the bench while we're doing this. You don't put your cap down, you don't put the tubes in a rack, everything's done in your hand. So, you'll hear me refer to clean tube and dirty tube. The clean tube is the tube with the fresh media. It's sterile, I know it's clean because it's clear. That is going to go between my first and my second fingers, just like this. The dirty tube, which has the species I'm taking and transferring, is going to go in my next 2 fingers, so it makes a V, kind of like this. Again, it feels very awkward, but you'll be used to it soon. The other thing I want to point out to you is these caps are not watertight. So do not tilt the tubes Because if you do, you'll get a spill and you'll have bacteria dripping down your hands. So always leave your tubes as upright as possible. Don't get distracted and tilt them around because you'll have a spill. And then you'll wonder why your hands are wet and they're covered in bacteria and you have to clean. Okay, which is fine, but let's try to avoid it if we can. So you have this claw situation going. You sterilize your loop. 10 seconds fully in the sterilizer once it's heated up to orange hot inside, which most of yours should be by now. It should be glowing orange inside the sterilizer. That means it's super hot. It's going to take about 5-10 minutes to heat up if you forgot. So plug them in, turn them on to high. Don't worry, it's your first time. 10 seconds. We are never, ever, 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 ever going to do this because you will forget it. And then when you go to grab it, you will burn your hand quite severely. We don't want that to happen. So, your students are learning, it happens, everyone makes mistakes. If you find, oh no, I did this, your first instinct is gonna be to grab it, don't. Call me over instead. I have some surefire techniques to get it out of there without burning myself. But try not to do that. So 10 seconds in the sterilizer. This is now super sterile. Now I have to wait a couple of seconds, maybe 30 seconds or so. My general rule is just till I get bored because the loop is screaming hot. If I put it into my culture tube, I'm transferring from the dirty tube to the clean tube. I want to take the bacteria out of its crowded, dirty environment and put it in nice clean media so it can grow. Basically, if I put it in here now while it's screaming hot, I'll hear a sizzle. That sizzle is the sound of my bacteria dying. I don't want that to happen. Yes. So, also keep in mind there are billions of bacteria in one drop of this culture. So you're not going to kill all of it. If it happens, just keep going. But we like to avoid it as much as possible. I sterilize my loop and I don't put it back down on the bench until I'm done to prevent what type of contamination? Environmental. I shouldn't be talking while I do this, but I have to today. So what kind of contamination am I at risk for right now? Analyst, right. And I labeled my tubes to prevent what type of contamination? Sample to sample. That might be a quiz question where I tell you, you know, we're not going to blow on our loop. What kind of contamination would it be if I blew on my loop to cool it faster? Analyst. Environmental would be if I put it down. So we're not going to let anything be put down. So this is where it gets a little more complicated. Take it slow and you'll be fine. My dirty tube. I always take the cap off using my pinky. That way I know what tube is what. If you do it the same way every time, you'll never mix them up. Then I'm going to see how, like where I'm holding my loop, I have a little claw. I'm going to take off the clean tube cap there and I'm going to hold it like this. My hands are full. Now, I just need to dip my loop. If I'm still not sure if it's full enough, I can always touch it against the side of the inside of the tube because that's going to have the bacteria I'm trying to get anyway. And then I'm just going to dip the loop in the bacteria. You don't have to stir it. I just have a habit of doing it. It doesn't do anything. And then I'm going to transfer that loop full of bacteria and just dip it in the fresh media. And you can also stir it around, but again, it doesn't do anything. Then I take it out and I sterilize it immediately until it's on the loop. 10 seconds. Then I'm going to cap up. I know my clean tube cap was in those first two fingers and my dirty tube is here. So that is one aseptic transfer. Now, I could put this down at this point into the rack. I'm not going to, but I have another one to do. This is my nutrient agar. You can hold it like this so that the slant is up. See how the slant is up there? It makes it easier to inoculate. And then I use my thumb to brace it at that 90-degree angle. This is solid media, so I don't have to worry about it spilling. I only have to worry about this one spill. If you're unsure and want to, you can hold it like this. I prefer to do this. So that's the way I'll show you. I've been waving this around, so I'm just going to sterilize it again to be safe. 1, 2, 5, 6. About 30 seconds. I'm keeping my tubes closed until I'm ready to prevent what type of contamination? That would be environmental from the air. The air is part of the environment. Dirty cap goes in my pinky, clean cap goes in my lip block. And then I'm going to dip. And then I just want to use the amount of pressure like I would use to tickle someone's foot with a feather. So very light pressure and just kind of wiggle it down the surface of the agar. Sterilize my wound.
So I just have a question. Why we have to transfer the bacteria like into two tubes? Because I thought in the first one they already have the nutrient.
This is a different culture. Sometimes bacteria like to grow on solid media rather than liquid media. Some grow better on solid media than liquid media. Sometimes for certain experiments you want solid media, so this is giving you practice in inoculating both types of media you'll use this semester. Sometimes you use solid media, sometimes you use liquid. So after this exercise you'll have experience inoculating both. So this is an individual exercise. You're each going to do this on your own. Call me over if you need a cheerleader or support. That's what I'm here for. First, you are going to take, each student is going to take 1 tube of nutrient broth, 1 tube of nutrient agar, and you're going to put that in your rack. You're going to label it. Then, when you're ready and your sterilizer is heated up, you're going to take a tube of your bacteria, make sure to check the labels, and you're going to do a sterile transfer into liquid media and also into the solid agar. And that is the exercise. You can straighten that out before you. If your loop is all bent, you can straighten it out.
Also, one more question. So we put the bacteria, the same material, two tubes.
Yeah, but you do one at a time.
So this tube of bacteria to the fresh nutrient broth, and from the bacteria to the agar.
Yep. Yeah, you're never transferring from sterile broth to sterile broth because then nothing will grow. Yeah. Yeah, for the first row, so you were doing serration marcescens. Row 1 and 4 are serration marcescens. Take your tubes first, and then when you're ready, you can take the bacteria. Oh, I also recommend do not work over your nose. Have your nose off to the side like I do, so that if you spill something, it's going to get on your nose and we have to sterilize your nose. Drop it, yeah, you can't drop it. Yay, you did it. Great. I love it when things work on right space. We just switched to that system last year, so it's new.
One more question. You said you were eating.
10 seconds, so 10 Mississippis. Or if you say the word second, that's a second. Longer than a second. Here you go. You'll be walking around, see how you're doing. Then you can sterilize it, orange, hot, and it's ready to go.
This row is row number 4.
No, so row 1 is across. Row 2, across. Row 3, across. And then row 4 is all the way in the back. Across. The row numbers that those refer to are across. So this is your seat, you're row 2. Someone can E. coli from up here? What am I slamming? Nope. When everyone is done, I need all tubes back up here. It's actually a safety thing. I can't let anyone leave the lab for the day until all the strains are accounted for. So don't put them in your drawer. What's up, Kevin?
I'll try to make sure I don't leave it wrong.
Okay, so if you lose your way and you need to know what to label it,
Your
right up there.
initials. Yeah.
You said the solid media is,
Slant.
uh,
The liquid media is Bronx, so NA is slant. And RA is sorry. You're just doing regular sepsis or E. coli. If you're in row 2, you should be doing E. coli. Well, no, you have to get bacteria. This is the clean media you're going to be putting bacteria into.
I'm trying to label it, so.
Yeah, so you're gonna label it, your initials, and then you label E. coli, today's date, and then this will be, yeah, for both of them.
Today's March 7th.
Yep. Yep, you're setting up 2 separate cultures, one into the solid media, one into the liquid. And if you're waiting, you can practice your claw grip with the tubes just to warm up. So are you right-handed or left-handed? Okay, so you're correct then.
So you ready to areas with the different areas?
Yeah, you can just write NA for nutrient number. Yeah, that's correct. So now we'll just sterilize your loop. NB. Oh, but remember what I said, those tubes are not watertight, don't tip them, right? Yeah, straight up and down, those are liquid tubes. Even your solid tubes, there can be some condensation on it, so I recommend you hold all the tubes upright at all times, or as close as you can get. Because those caps do not hold liquid. 10 seconds. What's up?
Liquid into solid, right?
No.
Solid to liquid.
Bacteria into solid, then bacteria is liquid. You don't have bacteria yet. You need the culture tube, so you have to wait till someone's done. Remember, the purpose of this is to feed the bacteria. So this is clean, this is sterile, this is sterile. If you put this into this,
This is the fresh media.
both are the fresh media.
Fresh media, okay.
You need, does anyone have a culture tube?
Culture.
Yeah, someone needs to, well, we only have 4 for a class of 16, so you have to wait.
But I want to get a visual.
Oh, so, can I just borrow this for a sec? This is a culture tube observation or assessment. See how it's cloudy already?
So like what you heat up
The loop, yeah.
then from transfer from
The culture, which has the bacteria, into the fresh media.
which one?
Both of them, but one at a time.
Oh.
And this procedure is in your lab manual on page 29. Yeah, right?
I'm just trying to remember the steps. So
Yeah.
this is it, right?
So I always work with my manual out, even though, you know, I've been doing these things a long time, I always have my manual handy. It's something from my forensics days, you always wanted your manual nearby, because it's like, well, how do you know you did it right that time? Oh, because I always follow my manual. Then you'll be more confident that way.
I just know I'm someone like, if I'm following a recipe, I have to physically check off each thing, or else
Yep.
I'll do it in the wrong order, so.
That might be hard while you're holding
Yeah,
tubes,
that's
but
why I'm like, I have to get it right.
whatever works for you. Everyone's a little different. Okay, so you have a culture tube. What culture is that? Serration. Okay, good. You're all set. So you're doing it in the slant. Okay, good. Good technique. That was a nice streak of the liquid. Who needs SMR sessions? Yep, good. Bunch of people waiting for E. coli. So if you have E. coli, you're not.
I don't, I think you need to sterilize it.
You just need to put the same name here. SMR sessions, yes, speaking. Oh, this is fine. No problem. Who needs to reach behind you?
Someone with E. coli. Does anyone need?
Someone need E. coli? Okay, does anyone need SMR sessions? You look like you need SMR sessions. Did you inoculate her? Okay, so you're all. Anyone waiting on SMR sessions? Okay, then people who are waiting here, you can do SMR sessions. Well, just change the label. Yeah. Okay, that takes a long time to heat up. That's not going to be ready yet. It has to be orange hot. So the see how hers is orange hot inside? It's not going to sterilize anything. So it will look ready. You have it as E. coli, so we can just take the marker and toss out for your SMR sessions. Or you can take a little bit of alcohol and like toss it off. But for now. Just to keep things moving, if you are assigned E. coli and SMR sessions comes up first, change the label on your tube to SMR sessions. Once you are done inoculating, bring the stop tube with the label back up to this rack. And always hold it and pick it up. Excuse me, sorry. Hi. Always hold it and pick it up by the glass, not the lid. I saw you pick it up by the lids.
Because I drive it.
Always pick it up by the glass, because one day that tube top is going to be loose, and the tube's going to come falling down. You're going to have a big mess. I speak from experience. It's like when you see warning labels on things, like do not believe that the pool is on it. The tubes with bacteria go here. The other tubes go in your drawer. So you're going to put those in your rack of incubation in your drawer. All warnings I'm giving you are mistakes that I've made. Making those mistakes, one set regrets doing that one. Everyone makes mistakes. Still here. What's up? Yeah, those are going to go in your drawer in a rack and incubate over the week. So most of the stuff is here. It's going to incubate at room temperature, so we're just going to leave it in our rack so it can heat. page 13. Thank you. Angela? Laura? And Miriam. And Miriam. Once you sat across from each other, it would be very easy to get that name. Eva? We got it. Heidi? Louise. Nina? Hazel-Ann. She's gonna play. It'll take me a while. I'll get there.
Thank you.
Hey. Yeah, it has to be like orange inside. No, it's not orange yet. Like I said, it takes a few minutes, but the people might be aware. So what we can do is switch this over. For Seahawk versus orange, oh, leave it on, you'll need it later. But Seahawk versus orange inside. Yeah. That means it's hot, so be careful. It is working. It just, like I said, you just turned it on. It takes 5 to 10 minutes to heat up. That's why when, as soon as you come in the morning, we use it every single lab, turn it on, get it. Wow, get it.
Okay, one more question.
So today we're only using the loop. That has a little loop on it now. The needle we only use for one specific type of master master. Yeah, needle and loop. We're gonna use the loop today. So how do we feel? First procedure? Any questions? Yeah.
So when we open the cap of the tube, and is this contaminated the tube? Like, because I'm afraid like something inside that cap will fall into a.
It should, as long as there's not liquid inside the tube cap, it
Yeah.
shouldn't be a problem, yeah. You know, just by opening the tube, there's a chance for contamination. The idea is to minimize that risk. You're never gonna get rid of that risk completely. But by holding the cap the way you do, so by not putting it on the bench top, when I open it, I hold it down. I
Yeah.
don't flip it up because that will invite the microbes to kind of settle in there. And I only keep them open for as long as necessary. So we're minimizing the risk of contamination. The only way we could get rid of the risk of contamination completely is if I went into a sealed room where there's a clean suit and a respirator and totally closed everything off. And it's not necessary for the purposes. But it's not organisms. So things that are all considered, this is a biosafety level 1/2 lab. Most of these organisms are more opportunistic in causing disease, which means they'll only cause disease if you're like immunocompromised, or that's why we follow so many protocols. If you are concerned about your health or exposure to any of these organisms, I welcome you to talk to your physician about it, because I don't know your particular medical concerns. But we take precautions. We wash our hands. If you have large open cuts, that's the only time I'll advise you to wear gloves, aside from staining, to keep those covered. And by minimizing the amount of spills and lab accidents we have, we also reduce our chances of infection. There are things like biosafety level 3 organisms, which are things like tuberculosis, things that cause disease in most people. Those type of organisms you have to work with in a much more secure and closed off environment. This is biosafety level 1/2. We have a door, only authorized people get access to the lab. You're never in here unsupervised. You're wearing proper personal protective equipment. So, just a little tidbit. Okay, so, looks like most of us are done. If you're not done, don't worry. You'll be able to catch up later. It'll be fine. You have time to do. All this information is on the lab review slide in the lab. So the next exercise we're going to do is preparing a slide and a smear on that slide for microscopy. Now, what is a microbe?
Something small.
Very, very small. What do we need to see microbes? A microscope. Fortunately, we have plenty. So we can't just take a tube and say, okay, I'm gonna do this. It's not gonna work. We have to put it on a microscope slide. So that is what the process of smear preparation is. We're taking the microbe and we're sticking it to a slide so that we can add stain to it. And that's another good point not to wear your best clothing here, because we work with a lot of stains. And by definition, they stain things. You can see my lab coat is all decorated. I do provide gloves for the staining procedures. You are welcome to provide your own. I wear a size medium, so if you're medium, you're in luck. You can borrow my gloves. If you're not a medium, I don't have all sizes, but you can go, they sell them in like CVS. I buy mine at Costco in packs of 2. So if you guys want to team up, if you all wear the same size and want to just buy some, it'll last you probably more than an entire semester. But I don't provide gloves. I'm just letting you share them. If you happen to wear the same sizes. Because I have them. The only reason I wear the gloves is because otherwise my hands will be purple by the end of this lab. It's all, it's cosmetic. Okay, so we need to prepare a slide, and then we're going to go through staining procedures. We need to stain the bacteria so that we can see them under the microscope, because most bacteria are colorless by nature. So if we put something colorless underneath the microscope, you might see the edges or like a shadow, but it won't be super, super clear. So we add dyes to add contrast to the specimen. In order to dye the specimen, we need to put it on a slide first. So now I'm going to show you the procedure for making microscope slides. And again, this is something you'll do a whole bunch this semester. I like to work over a paper towel. So since it makes it easier, personal preference. I am going to make 3 slides. You are only going to make 1 today. We'll do it again. So the three species you'll be looking at under the microscope today are E. coli, Staphylococcus aureus, or B. subtilis. And we're going to do it in rows. That way you only need to make 1 slide, but by going to your neighbor's microscope, you'll get to see everything. So, if you are in row 1, you are going to make a slide of E. coli. If you are in row 2, you are going to make a slide of Staphylococcus aureus. And you see where this is going? If you are in row 3, you can do Bacillus subtilis. And since the 2 of you are my only row 4 people today, pick whichever one you want. Whichever one's available at the time, your choice. E. coli, Staph aureus, or B. subtilis. We are not using the liquid cultures for this one. Instead, we are going to make our slides from solid agar slants. The reason is there's a little more robust growth on that agar slant, so you're going to have more material to put on your slide. I am going to start by making 3 slides, and again, you're only making 1, so I'm going to take these 3 tubes over to my working area, so they're ready for me when I'm ready. But first, I'm going to explain a little about what the procedure is. So we want to make a slide to look at under the microscope. we have to put the bacteria on the slide. And we have to get it to stick. Because otherwise, when we add this nice liquid dye we have, it's just going to wash away. And then when we look under the microscope, we won't see anything. So it'll have been pointless. So we have to aseptically transfer, using the techniques you just learned, of the bacteria to the slide. Since we're going from a solid medium, we're not using liquid this time, you need to put a drop of water on your slide. So you start by adding a drop of water. If you have a liquid culture in the future that you want to make a slide of, you don't have to add the water because it's already liquid. We add a drop of water to the slide, and then we're going to place some bacteria, a loopful, on the drop of water. Then we're going to spread it with the loop to make a thin film. And then the last step is we're going to put it on the slide warmer. And the slide warmer does something very important. The slide warmer will do two things. It'll dry the slide, and it'll also do something called heat fixation. It's going to heat fix the slide. So heat fixation is the act of sticking the bacteria to the slide so it won't wash away. So, heat fixation does three things. All we need to do to heat fix it is heat it on your slide warmer until it's dry. They're hot, it'll do enough. So heat fixation first kills the bacteria. It makes holes in the bacteria so that the dyes can penetrate better. And the third thing it does is it sticks the bacteria to the slide. And the reason this works is because it causes the proteins in the bacteria to do something called denature, which means they unfold. When they unfold, they tangle up together and stick. And that forms a really strong adhesion to the slide. The analogy I like to use is an egg. And I know eggs are really expensive now, so I don't know how many eggs we're all cooking. But if you put a raw egg in a frying pan, think about it. It slides around. You can remove it really easily into a cup just by tilting the pan. It'll all come out. But if you forget to add oil and butter and it's not a non-stick pan, and you start to heat up that egg in the pan, it changes, right? The egg white becomes clear, the yolk solidifies. That's because the proteins are unfolding because of the heat. They're denaturing and they're tangling up. Is it easy to get that egg out of the pan? No, you pretty much ruined that pan. It's going to stick there. You're going to have to soak it. It's going to be a whole production. That's what's happening on the slide warmer. The bacteria is sticking there, and we want it to stick there because we're going to do all these different washing steps to add dye so we can see the bacteria better under the microscope. So, I have 3 slides here. This will be my Sharpie. And as you can see, I never work over my notebook because I don't want to get bacteria on it. For the slides, you're not going to keep them week to week. It could be a little more casual with how you label them. I'm making 3 slides just so I have examples to show you. E. coli, I'm labeling EC, Staph aureus SA, and B. subtilis BS. And because I'm going from solid media to the surface of this media, you see kind of like fuzziness on it. It doesn't look clean like the one that you just inoculated. This is what the bacterial growth looks like. So you can see that. And you can see there's some liquid. The tube. The liquid is actually the byproduct of metabolism. these get low, we have refill bottles at the top of each sink. It is your responsibility to refill them. So they're full for the next class. Everyone does this, it's how we keep things moving. So if you need to add a drop of water to the bottom, you can come up if you need to see. So 1 drop. Again, you're only making 1 slide, so you only need to do this one. So I have my plant here. I'm holding it upright this time because there's liquid in it and it's already grown. Sterilize my loop. Because it was sitting on the bench, it may have picked up some environmental contamination. I'm not going to wave my loop around in the air because I might pick up microbes from the air. I'm not going to blow on the loop like I would some hot soup because that will get the microbes from my mouth on it. We don't want that. So once it cools a little, I still use my pinky to take the cap off. Then, I'm just going to use very light pressure. If I'm not sure if my loop is cool enough, I just touch it to the inside of the loop and that helps it go down. Now, I'm just going to take a little bit of bacteria off the surface of the agar. I'm going to, again, use that same pressure like I'm tickling something with a feather. You don't want to scrape it because if you take the agar with you, that's what you're going to see under the microscope, not the bacteria. And agar looks really big under the microscope, so it's going to hide your bacteria. So you want to use really gentle pressure. Trust me, even a tiny bit that you don't see on the loop is going to be thousands of bacteria under the microscope. So you don't need to stress about getting it up and little bit. I can see that there's a little bit of bacteria on there. I like to move the tube once I've used it, so I know I've used it. And I'm just going to place that loop full of bacteria in the drop of water and spread it back and forth to make a thin film on the slide. And again, you don't need to be too rough or too fast with this. You'll notice the water get a little cloudy. That's because the bacteria is in it now. I'm going to sterilize my loop again to kill whatever's on it. And then I very carefully place the slide on the slide warmer and let it dry until it's completely dry. And that's going to be the heat fixation step. I'm going to do that for each of these. Whenever you're ready, you can start with yours. Again, make sure your slide is, your sterilizer's heated up. So now I'm going to do Steph Arias. You don't have to wait for me to do all 3 if you don't want to. But if you're not sure and want to see it again, sure.
And we only take it on the surface.
Yeah, so you can get closer and see what I'm doing.
Not enough to scrap.
No, you don't want to like scrape because then you're going to take the agar with you and that's what you're going to see under the microscope. Just a little gentle pressure. You'll see some of it just stays there. Don't worry, you'll still get enough. You share with the person across from you, or next to you. There's one for 2 don't touch it. Did you leave it in there by accident?
No, I just was supposed to clock it.
So what you can do.
Just place it right here.
When you're done with it, this one, I just need that one. Those can go in your rack, and at the end of class, you'll put it in your drawer. This one's a little better. What you can do, if you find the handle of your loop is getting too hot, one thing I like to do is take a paper towel. This usually only happens when I'm setting up a whole bunch of inoculations at once, which you will do later this semester. Take a paper towel, fold it over, and then make a little handle wrap. But if you do this, be very careful. Well, it may slip, but more concerning, because you're going to be hit by this, more concerning, you don't want to let the paper towel touch the sterilizer, because it'll catch fire. Yeah, we don't want that. There's a little fire left. That's my goal. We're good. So I'm going to prepare for the next
This one turns two. It's a loophole.
You might want to label it with your organism. Oh, okay, careful. Before you do that, Miriam, I'm just going to come over because I sense that there might be a problem over it. Hold it all the way back. It's too hot, but no problem. Next thing, when you're done and you're waiting, the next thing we're going to talk about is staining. We're going to go through a couple of staining fundamentals, and then I'm going to show you a simple stain with the negative stain on page 19. So once you make your slide, so you're going to use Steph Arias. It's not in here, so it's out, so you have to get it when someone finishes. There you go. See? Works out. Yeah, unfortunately, I don't have enough posters for everyone in the class except for you. So you have to just share.
I'm not gonna do that.
Oh, I know, I'm just letting you know. It's not on purpose. I wish we had bacteria for everyone.
I just want to make sure I don't mess up.
Of course, I appreciate being careful. Absolutely. Yes. Of course, maybe I have an answer.
Okay, but hold on. Why is there paper on your slide? No, I did not do that. Mine is frosted. So this would burn. This would catch fire. So it's okay that your water is clumped up as long as you put that. But you never have paper on this because it might catch fire.
You can put it back, yeah. I see lots
if you put on anything bigger than that or something.
Yeah.
But I don't think.
I do take a lot of bacteria.
Yeah. Yeah, it's gonna take a while, which is
Mine's
why
just like a drop.
you start and then I go. Okay. Looks like most of us are done. Maybe another minute while I take my cough. Feels like I'm gonna tape it to my hand one day. Oh my. Now we've accomplished step one. You've made your slides, or you're in the process of making your slides. Step two is we have to add dye to it so that we can actually see it under the microscope. So dyes add contrast to the specimen, so it's clearer under the microscope. If it was just colorless, we wouldn't be able to see it well. And remember, bacteria are so tiny that you might have worked with microscopy before in your other classes. One thing you might not have done that we're going to do today is use oil immersion. So 1,000 times magnification. We have to magnify really, really big to see these microbes. So, little fundamentals about staining. Every cell has a plasma membrane. It's its boundary. To be alive, you have to keep your outsides out and your insides in. It's one of the qualities of living. Very important. It's not only talking in philosophy. But plasma membrane is made of, what are these little guys I'm drawing here? What are our plasma membranes made of? Phospholipids, right. So we have a phospholipid bilayer. Bacteria, fungi, protists have them too. In bacteria, they're a little different than ours. We have cholesterol in our membranes. Bacteria do not have cholesterol. Fungi have something called ergosterols. They don't have cholesterol either. So while they all have plasma membranes, between the different types of microbes, they're each a little different. And you'll go over those differences in the week one lecture material. It talks a little bit about how the different classes of microbes, bacteria, fungi, protists, and viruses are different. That's something I really want you to focus on for this weekend lecture. And you'll see that in the slides. But for now, you have your plasma membrane in your bacteria. These little circles I drew here are the phosphate groups of the phospholipids. They have a very strong negative charge. For staining theory, this is important. We have two types of dye in this lab. We have basic dyes. And I don't mean basic in like the simple sense, I mean like acids and bases. A basic dye has a positive charge. So it's going to be attracted to the negative charges of the plasma membrane and stain the cell itself. So a basic dye stains the cell. We also have, if we have basic dyes, then we also have acidic dyes. Acidic dyes are negatively charged. They are going to be repelled by the plasma membrane, and therefore they stain only the background. So in today's lab procedure, we're going to do a simple stain that's a negative stain. A negative stain uses an acidic dye, so it's going to stain the background. We're going to use basic dyes a little later on in a different type of stain, but for now, we're using acidic dyes. We're going to do what's called a simple staining procedure. Simple, not in the fact that it's easy, but it is easy. A simple stain only one dye. It may be acidic, it may be basic. The important thing is you only use one. Doesn't matter which dye. There are lots of different simple staining procedures. What they all have in common is one dye only. Okay? So a simple stain uses one dye. From a simple stain, you can tell some things under the microscope about a bacteria. You can tell, first, its shape. Second, its arrangement. And third, its size. You can, yeah?
You said this is what the information you gave from one dye, right?
One dye, yeah. If I were to take the smears I just made and just put one dye on them and looked at them under the microscope, I would be able to tell their shape, arrangement, and size. And usually, staining bacteria and putting them under the microscope is the first step we take in identifying a species, because they have unique shapes and arrangements and sizes. So, in week 2 of lecture, you're going to be introduced to a whole lot of different shapes of bacteria. For this lab, I'm just going to focus on the ones you're going to see today. So just know there are more than the 2 I'm telling you. So for shape, some bacteria are round spheres. We call this round shape a coccus. If we have more than one, we call it cocci. C-O-C-C-I. Bacteria you see today may be rectangular or rod-shaped. We call that a bacillus. Bacilli, plural. Now, just looking at the names up here, can you tell me anything about the shape of any of those bacteria?
Number 3 is rectangle.
Number 3 is rod shape, yeah. It's a bacillus, says so right in the name. Olivia? Number 2 is round, right? There's coccus right there in the name. Sometimes they name things very logically, sometimes not. So, that's shape. Arrangement. You could be scattered all about like sprinkles on an ice cream cone. It's the best analogy I can give. Just, you threw them and they landed randomly. We call this a random arrangement. You may have an arrangement in pairs. We call this diplo. Not like the DJ, diplo. So these would be diplococci. These would be diplobacilli. You could also be arranged in chains. One after the other, like boxcars on a train. Chains we call strepto. Where might you have heard of strepto before, or strep? Strep throat, right? So, strep throat is an infection, it's a bacterial infection of the throat caused by an organism called Streptococcus pyogenes. So, from the name, we know it's round, we know it's in chains, yeah. Pyogenes actually tells you something else about it. Pyogenes means to make pus. Yeah. The throat infection makes pus. So sometimes you can learn a lot about a bacteria by looking at the name. Sometimes not as much. Like you may be wondering, well, we just went through all this. Where the heck does Escherichia coli come from? Escherichia coli is a rod-shaped bacteria. Usually it has a random arrangement. You can tell none of that from the name. Sometimes bacterial names are named after the person who discovered them. A scientist named Escher. Coli, what is that? What part of the body does that kind of sound like?
Colon.
The colon. It lives in your colon. Staphylococcus aureus. We'll get to the staphylo in a second. Staphylo staphyl, this only happens with cockeye. Bacilli can't do this arrangement. So please never tell me you have a staphylobacillus. That's not a thing. Grape-like clusters, staphyla. So Staphylococcus aureus, clusters of brown bacteria. You may be wondering the aureus part. The aureus part is kind of neat. You've taken chemistry? What's the chemical symbol for gold? AU. When you grow it on media, it takes on a golden color. Kind of neat. So throughout the semester, you'll see different names of things. Sometimes they make sense, sometimes just looking a little deeper in the name can tell you a little bit about the history of the organism or where it's found. So the name is very important. Okay, so these are the names of the different shapes and arrangements you'll see. We're not going to really measure relative size today on the microscope, but by doing a simple stain using one dye, you can tell all these things about the bacteria. And we'll be able to distinguish these different species by how they appear under the microscope. We will also later do a differential stain. A differential stain tells you all the same things as a simple stain, but then gives you a little more information. To do a differential stain, you will always have 2 or more dyes. The Gram stain that we're going to do later today is one of the most common stains in all of bacteriology. You may have heard in the news Gram negative, Gram positive. That's what it refers to, how it stains under the Gram stain. The Gram stain tells you a little bit more about bacterial cell walls. So you can tell shape, arrangement, size, but you can also tell what type of cell wall it has based on how it performs in the Gram reaction. But first, we're going to start with a simple stain. So we are going to do a negative stain. I am going to just use what I have up front. You will use the same organism you used already. So if you did E. coli, do E. coli again. If you did Staph aureus, do Staph aureus again. We are going to do a simple stain using an acidic dye, which is called a negative stain. The acidic dye I'm going to use is India ink. We have them in these little ampules up here. To do this staining procedure, again, I have paper towel. I had 3 slides on my slide holder, so I'm going to take one off now so I have room. Mine is completely dry at this point, so it's already heat fixed and ready for the other staining procedure. To do the negative stain, you need a completely new slide. It works a little differently than making a smear. This is different because instead of adding the bacteria into a drop of water, we're going to add bacteria directly into the dye. So, again, if you want to come up and see what I'm doing, you're more than welcome. This is also found in the lab manual on page 2. So, I have 2 slides here instead of one this time. One is the slide I'm going to put the bacteria on. The other, this is my spreader slide. So, what I need to do first, if I'm putting something from a solid media onto a slide, what's the first step in making a smear? Dry, well, yes, you always sterilize your loop, but add a drop of water. I don't need to do that this time. My drop of water is going to be the dye. So, we're going to share these throughout the class, 1 per row. To open it, you squeeze it until you hear that crack. That's a little glass vial inside breaking so the ink will flow. This is India ink. It has a negative charge, so it's going to stain the background of the slide. I'm going to add 1 or 2 drops of the India ink to one edge of my slide. So I'm not doing it in the middle. 1 seems to be enough, so that should be good. Sometimes they're a little dried out in my head. Use your judgment. And I'm going to sterilize my loop. I'm going to take a little bit of E. coli, again, very gently, and I'm going to put it in that drop, just like you did with your water. But instead of making a thin film all over the slide, I'm just going to go back and forth to make a line. See why. This staining procedure has an additional benefit in that because we're not heating the cells first, they stay in a more natural state. Sometimes heating makes things look funny. So So this keeps them more like they are in nature. And I'm just gonna put it in a little line in my ink. And then I'm gonna sterilize it. Now, don't be alarmed if the ink sparks a little in here, that's kind of normal. What I like to do is just shake the loop around so it gets any of that ash off. Then, if you enjoy ASMR videos on Instagram, you'll love this. You take your spreader slide and you put it right at the edge here until capillary action spreads the ink all over in that line. And then you're just gonna push it with even pressure. It's probably not the best one I ever did. It's okay, it'll still work. Until the ink spread over the slide. Then, since this has live bacteria on it, it goes in there right away. Then, I heat until that ink is completely dry. And that's your simple stain. So what I'd like you to do is do your simple stain with the India ink on your organism who needs E. coli.
You're gonna share that.
Yeah, well, there's more here, so just one per row. If the row has one, yeah, take your really hard. You need 2 slides to start, remember. And then you just set it to dry. And once you do, then we'll continue on with the gram stain.
You said E. coli, right?
You were in row 2, you're doing Staph aureus. And again, if you missed the notes I read on the board, these are all contained in the review slides on the board. Thank you. Gotta take off your laptop. Yeah, there's no ink in this anymore. Yeah, thank you. Oh, this is also not ink. If you read the label, this is oxidase reagent. We don't, yeah, it shouldn't have been in there. Yeah, don't worry, it's not harmful. It's just something we don't use until much later in the semester. Yeah, always look at your label. Did you drop it on your slide?
No.
Okay, good. I was gonna say get a new slide because it might mess up your results. When you're done, just look up. The next thing we're going to talk about is the grams. Always ask. So. Give that to your, pass it on to the next person. Does anyone need an NDA?
Just all messed up about this.
Well, drop the bacteria in the drop and then spread.
Here first.
No. Drop, then bacteria, then spread. That way. Yeah, that's allowed. The bacteria in and then spread. Okay. Looks like we're cooking.
How are you doing, Olivia?
Yeah, now you spread. That's a lot of ink, so just be careful. You put the bacteria in it already? Good. It's very satisfying when it fills up a whole slide. Perfect. Okay, good. It's cooking. Put your bacteria in. Now you spread it. Good. Exactly, yes. And then you're just gonna put it to dry. Good. So remember to throw your spreader slide directly in the sterilizer bin. Anything that gets bacteria on it that's still alive goes in that bin. Okay, good. Yep, put it in.
Shake it around so it sparks the ink, right?
If you find that there's still some ink residue left on the loop, you can shake it around inside so it gets off. But okay. So what we're going to do next is I'm going to take you through the gram stain theory. I'm going to show you the gram stain. And then you're going to gram stain your slide that you made the smear on previously that should be completely defaced. Once you do your gram stain, you're going to come up to me and I'm going to give you a microscope number. That microscope is yours for the semester. Don't wave it around, just right direct in the sterilizer. You're then going to, so if you have previous experience with microscopes and you're comfortable, you can start. But I have screens up here. I'm going to be taking everyone through the procedure together. So if you want to try it on your own first, that's fine. It's in your manual. But if you want to get a guided tour of the microscope, you can wait for me. And I'll just wait till most people are ready. Thank you. Yeah, you can return the India ink up here then when you're done. So the gram stain is a differential stain, which means how many dyes are we going to use? 2 or more. This one uses 2 dyes. There are 2 dyes as well as some other chemicals that we need to use to make this work. So I'm going to take you through the theory first by telling you what the gram stain differentiates between. It's a differential stain. It differentiates between gram-positive cell walls and gram-negative cell walls. So bacteria have cell walls, most. There are a couple of species out there that don't have cell walls, but we'll talk about them another time. For the most part, all bacteria can be divided into gram-positives or gram-negatives. There are something called acid-fast cells we'll work with next week. There are also something called mycoplasmas, which don't have cell walls at all. But you may have heard of them recently because this winter there was a large outbreak of walking pneumonia amongst people that is caused by a bacteria called Mycoplasma pneumoniae, which doesn't have a cell wall. So you need different antibiotics to treat it. But this is not a disease focused course. So all bacteria have a plasma membrane, as we previously discussed. Plasma membrane has a negative charge. However, most bacteria have a cell wall as well. And that cell wall is similar in function function to how it functions in a plant cell or a protozoan cell. It's designed to help the bacteria keep its shape and resist bursting from osmotic pressure. Remember, osmotic pressure is the inflow of water from the environment. So if water keeps flowing and flowing and flowing into a cell, eventually the cell will pop. The cell wall helps keep the cell together under those conditions, so it doesn't burst. So, two main types of cell wall. You have gram-negative and gram-positive. Here's your plasma membrane. In a gram-negative, just external to the cell wall, to the cell membrane, is a substance called peptidoglycan, and I'm going to write it over here. Peptidoglycan is the main constituent, it's the main portion, and I just totally butchered that spelling, I apologize. I can't write and talk at the same time sometimes. Peptidoglycan. As the name implies, it has a protein portion, peptides, and a sugar portion, glycan. So, peptidoglycan makes up most of the bacterial cell wall. Plant cell walls, does anyone remember what plant cell walls are made of? Starts with a C. Cellulose. Cellulose make up plant cell walls. Some protist cell walls are made of cellulose as well. Again, this goes back to your week 1 lecture, what's the difference between different classes of organisms. Fungi have cell walls as well. Yep, exactly. Chitin or chitin, I've heard it pronounced both ways. I do not judge either way. I still don't know what the accepted pronunciation is. It's okay. I've heard people pronounce it both ways. But fungal cell walls are made of chitin, which also makes up the shells of insects. So if you ever like step on a roach and hear a crunch, that's the chitin, I know. But peptidoglycan is what bacteria have. Archaea, which we also talk about this semester, have something called pseudopeptidoglycan, which is kind of like peptidoglycan, but not. Again, read all about it in week 1, before Monday. So, in gram-negative bacteria, there is a thin layer of peptidoglycan, usually only 10 to hundreds of layers. And I want you to think of each layer of peptidoglycan like a chain link fence. It's strong, it's helping the bacteria stay together, so it's holding everything in. Gram-negatives have a very thin peptidoglycan. Gram-positives, that peptidoglycan is very, very thick, having thousands, if not hundreds of thousands of very, very thin layer upon layer upon layer, chain link fence. Very strong. Yep. In the gram-positive, there are all these atoms that hold these layers together. And these are called cycloic acids. So this is something you find in gram-positive cell walls. Gram-negative cell walls have this thin peptidoglycan and then another, which is another phospholipid bilayer. So we'll draw a so you remember. And this is called the outer membrane. And this is a second phospholipid bilayer that covers the gram-negative cell. So when we're doing the next procedure, you can pack it up, but for now, oh, you have it?
For the bacteria?
Okay, you'll get that one. So the gram-negative has thin peptidoglycan in an outer membrane. The gram-positive has a thick peptidoglycan. By doing the gram-staining procedure, we'll be able to tell the difference between these two. We'll be able to tell which of those 3 species are gram-positive and have thick peptidoglycan, and which are gram-negative and have a thinner peptidoglycan. 2 of those are gram-positive, 1 is gram-negative. Just tell you right now. So, why is it important to know if something's gram-positive or gram-negative? Number 1, it helps you identify it. There are lots of staphylococci in the world. Some are gram-positive, some are gram-negative. There are a lot of bacilli in the world. It's just another way to tell them apart. The other reason is there's a very important antibiotic that works by weakening the peptidoglycan bonds. You may have taken this antibiotic. It's known as penicillin. Penicillin works by weakening the bonds between peptidoglycan chains. When this happens, the bacteria can no longer resist osmotic pressure and they burst. That's part of the reason penicillin works to kill bacteria. Bacteria can't survive and they burst. In week 2 in the video library, there's a short 30-second video where you can watch bacteria treated from penicillin pop. It's very satisfying. Pop, pop, pop. Gram-positives are generally more susceptible to penicillin because they have all this peptidoglycan that's a target for the drug. Gram-negatives have this outer membrane that protects the peptidoglycan. So sometimes it's not as effective against the gram-negative infection. Gram-negatives also have something on the outside of the outer membrane called LPS, lipopolysaccharide. Lipopolysaccharide can be very dangerous when it's loose in the human bloodstream because it can cause shock, septic shock. This is not a disease-focused course, this is just for your information, but know that gram-negatives also have LPS, which is unique to all gram-negatives. You may have heard of something to do with LPS called the O antigen. The O antigen is part of the LPS. You'll see it in your diagrams in week 2. You may have heard in food outbreak news, there's a recall sometimes on romaine lettuce or on hamburger meat because of a strain of E. coli called O157H7. The O in O157 stands for the type of LPS that's in the gram-negative cell wall for that strain of bacteria. Different strains of E. coli do different things. That one, if ingested by humans, causes food infection, foodborne infection, where bloody diarrhea and perhaps kidney failure result. However, it's a normal part of the cow's intestines. So, what's harmful for one species isn't necessarily harmful for another. But you can impress your friends at parties now because you know what the O in O157H7 means. And this is why I don't get invited to many parties. I'm going to take you through how the gram stain works and telling these apart now that you know a little bit about the anatomy of the cell. The stain starts out with primary dye just you start with the primary stain, which is crystal violet. And as the name implies, it's purple. It has a positive charge. So where is it going to stain? The background or the cell? Positive charge. Cell. Great. You don't have to be so quiet. So we're going to leave crystal violet on for 60 seconds. And I have my crystal violet here. Purple chalk. So the positive charge is going to work its way all the way down to the plasma membrane and stain both the gram-positives and the gram-negatives purple at this point. So after you add the crystal violet, you have done a simple stain. If you stopped here, you would just be able to tell size, shape, arrangement. That's it. But we want to know more. So the next step, we're going to add something called iodine. And the iodine is known as a mordant. The mordant stays on, this one stays on for 30 seconds. The iodine is going to form covalent bonds with the crystal violet to make each individual crystal violet molecule bigger. A mordant is something that helps the dye stick. So if you picture like a box with a hole in it, and you put something that's just the size of the hole in the box, and then it gets bigger when it's inside the box, it won't be able to get out of the box. Does that make sense? It's hard to visualize. Doing my best. But that's what's happening here. You put the crystal violet dye in, you wait till it's trapped underneath the peptidoglycan, and you make it bigger so it can't get out. Okay? Then, we're going to add alcohol. And we're going to rinse it just until it's pale purple. If you you add too much, it's going to rinse out the crystal violet even from the gram-positive cells. If you put too little, it won't be enough to get it out of the gram-negative. So it's kind of like a Goldilocks and the Three Bears situation. You want to get it just right. It takes practice. Don't be discouraged if you don't get it on the first time, but you'll be okay. So when I add alcohol, what that's going to do is it kind of destroys the outer membrane of the gram-negative cell. So that all that's holding the crystal violet in is this thin peptidoglycan. And the crystal violet says, no problem, and gets rinsed away, because there's not much holding it in. However, you rinse the gram-positive with the alcohol, there are hundreds of thousands of layers of peptidoglycan. It can't work its way out, so it stays purple. So if you stop the gram stain at this point, your gram-negatives would be colorless, and your gram-positives would be purple. But when we get like an environmental sample or an unknown sample, we don't know right off the bat that, okay, we can stop now because the gram-positives are purple. We don't know if it's gram-positive or gram-negative. So we have to do something called a counter stain. And a counter stain is the second dye we put on. So primary stain, we're going to put on a counter stain. That's going to stain all the cells that aren't stained by the primary stain. So our secondary stain or a counter stain is safranin. And this stays on for 30 seconds. And this is a red dye. Now, a lot of students get into the misconception that the safranin is somehow smart and knows to stain only the gram-negative cells. But the truth of the matter is, it's a dye. It's going to stain everything, including your hands, including your clothes. That's why we have gloves and lab coats. Think back to your elementary school art class. What two colors make purple? Blue and red. Blue and red make purple. So you have purple on the gram-positives, and you're adding red, it's just going to get more purple. So the safranin will stain the gram-positive cells, but you're not going to notice the color change because it's already purple. However, the gram-negative cells are colorless, so those are going to stain red. So if it stains red, it's gram-negative. If it stains purple, it's gram-positive. Good way to remember this, purple positive, both start with P. Gram-negative is red. If you call this pink, the mnemonic doesn't work. So gram-positive is purple, gram-negative is red. So I'm going to demo how to do this stain, and then you're going to do it, and then get your microscopes and have a look. So on your sinks, you have slide holders. You'll use those and use the sink for water rinses. And again, I wear gloves simply because I don't want rainbow hands. If you get dye on your hands, it will come out eventually. What I like to do is when I go home, I do a little sugar, and I give myself a little sugar scrub. Your hands off. So I'm looking for my crystal violet. I get all my dyes out first. Crystal violet, followed by iodine, followed by alcohol. Please note in your stain kits there's something called acid alcohol. That is not what you're using today. Make sure you only use the plain alcohol today. And safranin. So I'm all set up. You can count. You can also use a timer. That clock doesn't work. So I'm just going to use my watch as a timer. So the first step is crystal violet for 1 minute. All I do is add the dye onto the slide. You don't have to be precise, just cover the area with your bacteria, and you let it sit for 1 minute. Then I move the stain bottle out of the way, so I know I used it. Any questions so far? If you do, just ask. This is the longest part of the stain. It's a minute.
That's the purple.
That's the purple.
How long do you let it sit?
1 minute. 1 minute.
1.6 seconds.
A lot of science is waiting. Give us time to talk about life, talk about science.
Can we start taking our?
If it's heat fixed, you can take it off the slide warmer. Use your slide forceps though, because it will be hot. 17 seconds. Between each of the dye steps, I'm going to rinse with water, but I don't need to dry them off in between. can stay. And use the water in your sink. Okay. So now I just let the dye run off and then give it a quick rinse under the water. And you may see like a little clump of purple on your slide. That's normal. If you don't, that is also normal and okay. It doesn't mean anything's wrong. Next step, iodine, 30 seconds. And again, I'm not drying the slide in between. I'm just kind of squirting it on. And that sits for 30 seconds. Good time to make sure my alcohol is nice and filled, because that's the next step. And again, you're just doing the water rinse in between. I'm also going to get my bibulous paper ready, because that's what I'll need to dry the slides when I'm done. Nice. Now for this next step with the alcohol, all I'm going to do is rinse until it's pale purple. Your lab manual says rinse it till clear. In my experience, that is always too much. And you'll rinse the crystal violet out of the gram-positives as well. So what I recommend is maybe 7-8 squirts of alcohol. It's usually enough. Light to pale purple. So rinse the iodine off. And you can see, my cells are heat fixed to the slide. They're not going anywhere.
So I have a question. Before you put all these 4 dye in it, you use the one that we did in the previous?
You're going to use the smear that you did that didn't stain.
Didn't stain.
You didn't stain. So not the India ink one, the other one. So what I do now is I'm going to take my slide and I'm just going to rinse it 1, 2, 3, 4, 5, 6 until it just comes off like pale purple. Try not to drop your slides in the sink like I almost did. It's still coming dark. That's a little better. Okay. It's not an exact number, just till it's pale purple. Then you rinse it from the alcohol. Now in the last 30 seconds, yep, that's right. Next is my safranin. And I notice my alcohol is really empty, so I'm going to refill it in between doing safranin, 30 seconds. And you can see gloves are helpful for this. Not 30 minutes, 30 seconds. Any questions? So you're going to do this on the slide you made first. That's unstained. By now it should be more than dry. Final rinse.
pull the top off. Yep, and then you fill it up from there.
No, that's Purple Fusion. You want Crystal Violet. So there are other dyes in there that aren't used for this staining procedure. Make sure you grab the right one. You're missing Crystal Violet. So here's the Crystal Violet. There's only one for two of you, so remember to share your paint box. If anyone has extra Crystal Violet in your paint kit, please let me know. We're missing one. They get shifted around sometimes.
Oh my goodness. All the way.
Really, it was like right when it picked over to 34, but so once it's 35.
Yeah.
You go when you go to the conference in April.
I don't, he was like, you have to email me back in 24 hours, and I forgot, so.
I emailed him back more than 24 hours. He didn't reply back.
I don't
When
know.
I talked to the girl, his sister, she said that I will call even he told her. Also because I asked him for a recommendation. So I think that it's a nice gesture that I should go. Even though I'm so like. I don't know. Maybe second.
So if you have a hose
I don't know if I
on your water,
did.
just make sure that the hose is behind, like you have now, the slide holder. Because if it's in front of the slide holder, it's going to pour it all over you. Butter. That sink has been broken for years. Yes, after each step in the gram stain, you do a water rinse. So crystal violet 1 minute, rinse with water. Iodine for 30 seconds, rinse with water. Alcohol, rinse with water. Safranin, rinse with water. Dry. Put your phone away. Make it cleaner. Don't want anyone's phone to get hurt.
To the purple
I feel
pale.
like light purple. Maybe one more. And then I'll give it to you. I think that's, I mean, it looks like purple to me. I don't know.
And when you're done with your culture tubes, please remember to bring them back up. I am missing 1 V subtilis, 2 E. coli, and 2 Staph aureus. You shouldn't need the culture tubes anymore today, so it's okay to bring them back up.
That is a thing.
Yeah, I think it looks great. Cause I think,
Have a question?
yeah, I think it's okay.
Yeah, it's absolutely okay. You can't judge your slide until you look at it under the microscope. Sometimes I'll get a slide that looks like it has absolutely nothing on it.
Yeah.
Remember, these things are microscopic. You can't see them with our eyes. So try not to be judgmental over your slide until you look at it under the microscope. It may look like nothing's on it. That's fine. Their bacteria are super tiny. It just means you did a really good job of spreading them out.
Yeah.
You ready? Okay, why don't you take microscope number 1 then? So when you take it, you're going to grab it with 2 hands, take it back to your bench, and then come back for a pour. If you're at the point where you have your microscope and your ink, your India ink stain is heat fixed, if it's all completely dry, you can shut off your sterilizer and unplug it for the day. If everything's all heat fixed.
Professor, I didn't do another slide.
Why?
The one I went to the bathroom. So should I do it now before taking the microscope or?
No, you can look at someone else's today. Yeah. We'll be doing that technique again next week, so you'll get another chance. I just don't want you to miss out on the lab activities because you're starting. So keep the cover with you. Keep the cover with you, yeah, because you'll need to put it back on. There are little cubbies underneath you can stick it in. You ready? Yeah. 3.
I just really dry.
Nina. 4. This one didn't have a cover. That's okay, some of them don't. If your microscope has a cover, keep the cover with you and put it back on when you're done. If it doesn't have a cover, you can't do that, so it's okay. Take number 5, please. Okay, you ready? Yep. 6. Remembering the last one. 7. Alrighty.
couldn't tell
Miriam's
you.
microscope. So
Oh.
Miriam, take that one. You take number 12.
I thought another very. I'm number 12.
This is where making track of microscope tubes in case there's a problem I can get you on. And Miriam will be number 15. So once you put it down, you can go back and get a table. Yeah, the tables are separate from these places. So we're gonna start by looking at our gram stain slide. Left and see. So what you're gonna do is take your microscope, make sure the oculars are facing you, plug it in using the power cord, and then they turn it on. So there's a switch on the side of the back. You can turn the light up using the switch on the side. If you're new to microscopy, I'm going to be doing a step-by-step demo. Some of you have already started, but that's okay. So plug in your microscope, get it turned on. And then I want you to put the stage, which is the part your microscope slide will rest on, in the lowest position using the little wheel on the side, which is the first plus. And you're always going to start with the red objective in place, so other way. All the way down. The red objective is a working position. We always start with the lowest power magnification, so we'll look. Yeah, you're also using so yeah, you only use the force it does on this. Never use the force it does once you lose all of this. You might crack the slide. Your slide. So, just waiting. Ready? Plug in your microscope. We're going to start with the objectives. And your objectives are on this little carousel here. The 4x objective in the active position. And just quietly listen for a second. Hear that? You want to hear that click. That means it's fully engaged and in the right position. And you're going to start with the shortest objective, which is the red one. That's your 4x objective. That means you're getting 4 times magnification from the objective, and we also get 10 times magnification from the eyepieces. So our total magnification is the ocular multiplied by the objective. So that's going to be 40 times magnification. You might get a question like that on a quiz. What's the total magnification if my oculars are this and my objectives are that? So remember, you just multiply those two numbers together. You're going to start by moving your stage to the lowest position using the big wheel on the side, which is your course adjust knob. I have to turn my wheel towards me to get the stage in the lowest position. That's your loading position. If you're not ready yet, don't worry, it'll catch it. Take your slide. You're going to clip your slide in with the bacteria side up, very important, into the slide holder, which is that little metal clip on the stage. And you want to make sure that it's flat against all corners. You don't want it loose or wiggling around in there. And you want to make sure it's flat with the stage. I'm going to run around and check, because this is often the cause of most student problems with the microscope. You're in the position. Very good. Back to you. If you're not up for this yet, no worries, we'll get you there yet. Yeah, good. Good, we're gonna start actually with the gram stain slide because the negative stain slide, I'm just gonna warn you, is super hard to focus. So we'll start with the easy one. Oh, so you need a different cord, because this one, yeah, this one has a different adapter. So yeah. Can I have that one? I don't even think we have that microscope here anymore.
Yeah, that's the one.
Okay, thank you. Okay, now I'll check on this side. I just want to make sure everyone's slide is properly in. Good. Good. Here. Why? Yeah, tell me more. What are you? I don't know. Why do you also label water? Oh, you always label it with the bacterial lead. It's okay. No, don't judge your slide until you look at it under the microscope. Good. Okay, great. Okay, so once you do that, and your objective is in the 4x position, you're ready to start focusing. So my rule here is, if you drive a car, I hope you always look at the road. You don't look at texts, you don't look anywhere else. You're always looking in the direction you're going. It's how we stay safe. A microscope is not a moving vehicle, but the same rule applies. If you're driving the microscope, you're always looking in the objectives. Very important. Don't focus without looking, because you'll miss your stop. So, what you're going to do, you have two knobs on the side. You have a tiny knob, which is your fine adjust, and then attached to that knob is the bigger coarse adjust. The coarse adjust causes the stage to go up and down quickly. The fine adjust causes smaller movements. So we're going to start by looking in the oculars and bringing the stage up using the coarse adjust until a picture comes into display. Now, this picture is not going to look like bacteria. It's not going to look like anything identifiable. You're not going to see bacilli or cocci because the magnification is too low. Instead, what you're going to see is a pink or purple haze on the slide. If you're not sure if you're focused on the slide properly, you can move the stage using those two little knobs attached to the stage, which are the X and Y axis controls for the stage. And if the picture moves, then you're looking at the right thing. If the picture doesn't move, you're probably focused on the lens below and keep trying to focus it. So what I'm going to do now is I'm going to look in the microscope and I'm going to use the coarse adjust to bring the stage up until I see kind of haze or a little bit of dirt or a little bit of schmutz. Schmutz is a tech. And this is what I'm seeing when I'm in focus. You can look up on the screen. I'm just going to do the white balance. Doesn't look like much of anything. It's just some stuff on the slide because I'm at a very low magnification. But I know I'm focused on the slide because I move my slide and the picture moves with me. So give it a go. Well, you're already on a higher magnification.
Oh, I did it the whole time.
You did it at 4x already?
Yeah.
Okay. Because you're focused at 4x, 10x. So you went ahead. So I warned students that sometimes the 40x doesn't work so well, but yours looks fine. So you could just hang out until we're ready to go to oil. Right there? Sure. So there's this wheel on the side. That's the light control. So that beam of light is coming up. That'll make it a little easier too. If anyone needs to be checked out, let me know. Once you get a nice picture at 4x, you can move to the 10x. So the 10x objective is the yellow one. It's 10x from the ocular, 10x from the objective lens for 100 times total magnification. And this is what I see at 100 times total magnification in my slide. Still not really identifiable. There's a rule though. Once you move past the scanning objective, which is the lowest power objective, you are never to use the coarse adjust again. What happens is I want you to notice how much working distance when you go up in magnification there is between the slide and your objective. It gets smaller and smaller and smaller. If you use the coarse adjust, With that shorter working distance, you're going to move the stage and the slide up into your objective, and you're going to crack it, which is going to result in glass flying near your eyes, which is never a good idea. So you only use the fine adjust. Fortunately, these lenses are what we call parfocal, which means as you go up in magnification, you should only need to make tiny adjustments to the focus to get a nice picture. If you're doing more, something's probably gone wrong, you go back to the previous magnification and try it again. You can always go back to the 4x objective. So this is something like what you'll see at 100x. Once you get a good picture at 100x, you can move to the blue objective, which is the 40x objective. And I will warn you, these might, why don't you get your microscope, Erin? No, go ahead. That one you can follow along. It's okay. Thank you for being so considerate. So the 40x objective on your microscopes may look really not great because these are student microscopes that have been in our labs for many, many years. And what happens is students don't clean them properly, and the 40x objective gets caked with oil, which is something we try to avoid. So things might look really dirty at 40x. So if you don't get a good picture at 40x magnification, 400 times magnification, just go back to the 10x and focus there. And then we'll go to oil immersion, because you might spend all day trying to get a nice picture at 40x. It might not happen just because the lens is so dirty. So it's not you, it's the microscope. And if you have trouble at this point, you can call me over and I'll have a look and see how we're doing. But you should have a nice picture at either 100x or 40x. I'm going to show you on my screen what 40x looks like. I'm always looking where I'm driving. I'm just looking. See, even on the instructor lens, it's a little dirty looking, a little cloudy. That's okay. I can still start to see what's going on the slide. So this was my bacillus subtilis slide. And now, I don't know where my oil went. I had to get started. Just gonna look for my oil. Put a bar. Okay, anyone need help? Okay, give me that. And then I don't want to put this on your lap, so I just have to move out of the way. Okay, so yours just requires a little bit more arm strength than this. Here we are. Sometimes it's just a patience game. You just gotta keep trying until you get it. There we go. No problem. And that's kind of what you should be seeing. It doesn't look like much, it just looks like there's some stuff on it. Yeah? Yeah, there's a pink piece, but we can get this better. Sometimes it's easier to do a part of the slide with less stuff on it, because then you're looking at more than just like a piece. It's easier to focus when you have edges. Yeah, because you've got a lot of bacteria on the slide, which is good, but also can be bad when you're trying to focus. Here we are. You're welcome. Yeah, you know it's there because it doesn't move when you're on your slide. So when you clean up today, you're going to take some lens paper with some alcohol and clean that extra well. Like I said, these are very well loved microscopes. Okay. So when you're ready for oil, what you're going to do is you're going to move the objective so you're between the 40 and the 100x. So it's not clicked into place. Then you're going to take your oil and you're just going to add one drop to the slide. There's no space in between, and that's what it has to be. You shouldn't have your objective all the way up here and your slide all the way down there at this point. Because what the oil does is it's refraction. What happens is it's bending the light so it gets into the tiny hole on the 100x objective. So it needs to touch in order to work. If you call me over and tell me you're not seeing anything and I see your oil immersion objective all the way up here in like that much space, that's why. Now, that being said, you're at 1,000 times magnification now. So little movements are really big on that small scale. Like a little adjustment is going to go a long way. So when you focus, I'm going to move this back to my eyes for a second. When you focus, you have to be looking the whole time and just go very slowly with the fine adjust. Because it's going to come in and out of focus like that. And you might miss it if you're not looking or going too fast. If you find yourself going in one direction for a long time, try going in the other direction for a little while. If you get totally lost, once you're on the oil immersion objective, you can always go back to the 4x objective, but you can't go back to any of the other objectives because you'll get oil on them. Okay? So, this is what I see under oil immersion. And now I can identify that bacteria pretty well. So this is gram-positive or gram-negative? It's gram-positive because it is purple. It is screamingly purple. Is it rod or round? It's a rod shape. This is a rectangular bacteria. Is there any special arrangement you see there?
Strains.
Chains. So it's rod shaped, so what does that mean? What's the science name for that? Bacilli, yeah, it's bacillus. And it's in a chain, so it's strepto, yeah, the arrangement name. And so I have gram-positive streptobacilli. That corresponds with Bacillus subtilis. So this is what, if you have Bacillus subtilis, this is what it should look like on the. So I'll go through each of the organisms just in case you don't get time to visit your neighbor's microscope or you have trouble. These pictures are also found on the review slides for this week. And Olivia, I took your oil. And if you're having trouble, by all means, call me over. But if you want to look at the screen for a second, I just want to show you how quickly it goes into and out of focus. I'm moving it out of focus now. I moved it like less than a 30 seconds of a turn. And then it goes back in really quick. So you got to keep your eyes on the oculars when you're doing this. See, lots of streptobacilli.
You said it's 110, right?
So if you do the oil, to put the oil on the slide, you're going to move it between 40 and 100. And then you put the oil down, and then you move it into place. So you need to put the oil down. Yeah. So it's 12:01 now. We have about 15 minutes left before we have to start cleaning up. You might not get to look at your negative stain today. That's okay. We're going to be doing a similar procedure next week, or you can store your slide in your slide box for next week.
You said between the 40 and 100.
But yeah, but this is your focus, so it's okay. So we go back, we go to 4x. Okay, looks good so far. Now we want to go to oil. So you move it between 40 and 100, and you just put a drop of oil right where the flag is coming up, and you will spin it 100. Did you focus at 4x already? Which way are you going? Yeah, you can always move the stage. You just can't use the course adjustment. Did you put oil down?
Yeah.
Okay. The 100x, the one with 100 written on it. So not the blue one. That's what it's there for. The oil should be on the oil immersion lens. That's where we want it. So, we have this slide. Let's start at the beginning. We're at 4x. Lots of light, good. So we'll move this around a little. See a little there. Yeah, I know. It's like you see just like these random stuff. There is stuff on your side. Have a look. Oh, it looks great. So this is, I'm guessing, E. coli. So, perfect. Great job. So, what you're seeing is it's red. So it's gram. Zero is positive. Red is gram negative. Is it rod shaped or round? No, it's rod shaped. It's kind of like a marshmallow shape, but yeah, those are rods. Those are definitely rods. And they're in like a random arrangement of sprinkles on an ice cream cone. And that is what you expect to see for E. coli. So great. So Heidi over here has a great example of E. coli if you want to come have a look. She has gram-negative rods in a random arrangement. She did a very nice job focusing too. So now you're going to go up to the 10x and you're just going to find it up. Do the same thing as best as you can. However, since you have so little on the slide, it makes you a little harder. Yes. So we've got a little close here, which is good. So you really have to work that out right here, because
No.
it's so little on the slide.
If you want to look at mine, I got pretty good focus on E. coli.
Mine is just a little on the slide. I'm not sure if I'm going to draw a line on the slide, and then I focus to that. And that way I know I'm in focus on the slide, and then I move it over to look for stuff later. So if you get really frustrated, you can always focus on a line. Yeah. Oh, you won't know with that magnification. But I'm guessing you have scalp artists. But, so now you did it at 10x, and it's great at 10x. Now you go up to 40, and there we go. There it is at 40. And now you're ready to put a drop of oil on and move it to 100. We're going to have to clean that really well. I did not know. Okay. So there we go. Oil on there. Let me know next time. Okay. Yeah, looks awesome. I'm guessing you have stuff about Staphylococcus, right? Okay, good. Okay. So what you're seeing, is it purple or is it red? It's purple, so it's gram positive. Is it round or rod shaped? It's round. It's round. They're round. They're round. That's what round looks like. And they're in clusters. So that makes sense given you have a very nice example of Staphylococcus aureus. So that's what it should look like. Have you done that? Do you mind if I work? Ooh, nice Bacillus subtilis. Yeah, so once you get a picture, you can marvel at it for a while and say, good job me, and celebrate, because it's exciting. This is your first microscope slide. You can take a picture of it if you're good at doing that. I am not. But then visit your neighbors and see what theirs look like. And you're all doing a really good job. Okay, so you have pink, well, red, so this means it's what? Nope. Gram negative. Look how purple, yeah, that means you might not have added enough alcohol. But purple, that's the purple you're looking for it to be positive. That's red. It's rod shaped, yeah, so it's gram negative, it's rod shaped, and it's random. That's rod. So the E. coli rods aren't like perfectly rectangular like this one is, like they're not elongated. The E. coli rods are a little squished. So round bacteria, you only have one species of round bacteria, the Staphylococcus aureus. It's very round, like a beach ball round. The E. coli you're seeing looks more like a marshmallow, it's kind of squished. That's still rod shaped. So, something, yeah? How are we doing, Nubia? It looks great. What are you talking about? Because it's round, it's purple, it's in clusters. Good job. So we have a Staph aureus example over there too. So visit your friends, make friends, exchange contact information for study groups or WhatsApp chats. Okay, I'm not seeing anything either. Let's see what we got. Because you definitely have stuff on the side, so it's probably just a simple one. So I got it nice and sharp here. So now I'm gonna. Sometimes the 100x is just a patience game. Oh, no. It's not just you, but I got it. It went in now so quick I almost missed it. There you go. Okay. Looks great. No, they're not round. They're rods. They're rounded rods, but that's what, like I just said, that's what you should see with E. coli. This is B. subtilis. You have really long rods with B. subtilis. With E. coli, everyone's judging E. coli's shape so much. So B. subtilis is gram-positive, really long streptobacilli. Okay? Staph aureus, really round, gram-positive clusters. E. coli is kind of marshmallow shaped, but it's still a rod. It's definitely not round like Staph aureus. And this is gram-negative. And this will be in a random sprinkles on an ice cream cone arrangement. So that looks perfect for E. coli. It does. Okay, you're good. I get very excited, I'm sorry. I understand people, yeah, that's good. Yeah. Looks good. So this is Staph aureus? Yeah. So you'll notice some of the purple is lighter than others. That's just a sign that you're just on borderline of having added too much alcohol. But you did. You saved it. If you notice you have pink and purple mixed together, like in the same field of view, not it's purple on one end just because the alcohol didn't hit it that much, but in the same field of view you see pink and purple, that's sometimes a sign that you over-decolorize. The rule is, in the same field of view, if you see really clear purple cells, call it gram-positive, because you probably just added a little too much alcohol. If you see no purple, then it's gram-negative. This takes practice, so have a look. If you've looked at everyone else's, you're good. You can try and look at your, if you want to take like 5 minutes and try and look at your negative stain, you can do that as well. I will tell you, and I'll show you up here, it is a difficult focus, but you all did such a good job with focusing under oil that, I don't know, I don't think you'll have a problem with that. But I'm going to just show you here what it looks like.
Well, I have a slide.
So, this is my negative stain. outside. See, see those little holes or spaces? That's where my bacteria are. Now I'm gonna go.
I think it looks like
It's so small.
it looks like little round. It looks like little round dots.
Oh, right.
Yeah. Or I think it could be clusters, but I don't know.
Yeah, like cluster.
This is what I mean by this is a hard one to do. So what you're seeing is the background, which is where the ink is. And then you see how I have little areas of those stains in place. That's where the cells are. And remember, this is a simple stain, so all I can tell is the morphology and the arrangement. So the shape and the arrangement and the size. My E. coli, this is E. coli,
The second
are those little, I wish I had a pointer, but I'm very short. So there, those kind of scattered. And that's all you should see. That's all it is. I find the gram sheet a little more satisfying, but that's what negative stain is. Next week, we'll use the negative stain in combination with another dye. Using 2 or more dyes is what type of stain? Differential. We're going to look at capsule, which is outside of the cell wall, by using lithium ink as a primary stain and then crystal violet as a counter stain to stain the cell within those little white spots a different color so we can see if there's a capsule surrounding the cell. So if you didn't get a chance to do a good negative stain today on this slide, you'll get another chance to practice it next week. With the gram stain, we're not going to do a gram stain next week. Next week we're going to do an acid-fast stain, which is a third type of cell wall. And we're going to do an endospore stain on B. subtilis, where we'll see a special survival structure that it produces when it serves. And those spores can survive for tens of thousands of years in the environment. They're super sturdy, radiation resistant, all that stuff. So we'll stain those next week. If you're done looking in your microscope, it's 12:17 now. We have a few more minutes, but we like to stop a little early to clean up. So clean your microscope. You are going to take the stage and again move it to the lowest
These are by 10,
position.
and then
Put the 4x objective back in place. You can discard or keep your slides in your slide box, your choice. The slides are cool, so you don't need to use the slide process. Just use your fingers. So for the oil, what you're going to do, so the slide has oil on it. If you already looked at it, you might as well just discard it because I'm saying if you didn't get to look at another one of your slides, you can keep it. The oil tends to make the stain break down, so it doesn't last long once you put the oil on. You're going to take some lens paper. You're going to take some alcohol. So you take the lens paper, which is the tissue paper like one, and you rip out a page. And then take the alcohol. First, use the alcohol to clean the off. Then, use the alcohol to clean the 4x objective. Then the 10x objective. Then the 40x objective. And save the oil immersion one for last. So you don't spread the oil anywhere. Once you do that, you're going to move the 4x objective back into place. Make sure your slide is no longer there. Put the stain in the lowest position. If you had a cover, put it back on. For your pores, into your drawer. You're gonna wipe down your bench with disinfectant, unplug and turn off your sterilizer, and tuck your chair in. You then wash your hands, remove your lab coat, and have a fabulous weekend. So I'll see you next week. Congratulations on your first lab. You all did a great job. I saw so much good focusing and technique that I'm excited for the rest of the semester. I think it's going to be great. I will see you all on Tuesday morning. Don't forget to do your first annotation on the week 1 lecture slides before that. Okay, and all the tubes have come home. Thank you very much. And I found my other top too. Oh, yes. Thank you, Lena. I'm sorry. Yeah, wipe down your pens and return them. They go right in the old pens up. Your tubes and your racks go back to your drawer. The lectures. And the syllabus, yes, there is a syllabus annotation assignment credit that you used to making annotations. You make 2 annotations on the syllabus for 5 points. If you haven't uploaded your safety sheet yet, make sure you do that by today. I think most of you have now. Yeah, for the annotations, yeah, you have to make, for the lecture slides, you have to make 1 before class on Tuesday, and then the rest are due on Wednesday. So you have to make 3 annotations and respond to a classmate's annotation for the assignment to be complete.
What does annotation mean?
So, have you been on Brightspace?
Yes.
So you click on where it says hypothesis assignments. So the annotations, there are some examples of what you could do as an annotation. You can ask a question, you can define something and add your own analysis to it. You can post a resource, but you have to add your own. You have to say how it's relevant. You can't just say, here's a video. You have to say, here's a video that explains this, and I like it because.
Something related to what we
The slides, yeah.
next week, next lecture.
Monday, yeah.
Monday.
Tuesday.
Tuesday.
Yeah, I have another section on Monday, so I apologize if I say Monday
So
instead.
there isn't anything we submit for live?
No, you have a quiz next Thursday. It's Thursday,
We
right?
also have to comment on other people annotations
Yes.
as well. And
One
only
other person, yeah. But as long as you make 1 annotation before class on Tuesday, you're good. If you choose to do all of your annotations before Tuesday, that's fine. One thing though, if you do 1 annotation before Tuesday, you'll see yourself get half credit. That's a good thing. That's just me telling myself that you made the 1 required annotation before class. I update all the grades to full credit if you did the required number of annotations on Wednesday evening. If you need it, keep it. Are you sure this is not even, this is my own, it's paper, keep it. And if you have any questions at all over the week, or you're not sure what you're supposed to be doing, or where to find something, do not hesitate to send me an email. As many of you know, I answer